Hey everyone, in this video I am going to describe a technique called northern blotting. So northern blotting, what is northern blotting used for? Well, I used to do this a lot during my PhD. It is a technique used to detect a specific RNA molecule in a sample of RNA. Okay, so a technique to detect a specific RNA molecule in a sample of RNA. So let's say maybe the best way to introduce this technique is an imaginary experiment where we need to use northern blotting to test the hypothesis. So let's say we have a hypothesis that this gene, CSA, now this would be a gene for the CSA protein that is involved in transcription-coupled nucleotide excision repair. This is a technique we talked about in a previous section of the course. So let's say CSA messenger RNA levels increase after exposure of, let's say, cells to UV light, which might happen, right? So UV light causes pyrimidine dimers. Pyrimidine dimers are removed by nucleotide excision repair. Transcription-coupled nucleotide excision repair is one of the two forms of that genome repair, of that DNA repair process. So so maybe CSA messenger RNA levels increase because maybe the cell needs more of that protein if it senses or is able to detect that DNA damage levels are high throughout the genome. So let's say this is our hypothesis. It might not be true. And if we wanted to test that hypothesis, well, we could use northern blotting. So how would we set up this experiment? So we would need 3 batches of cells, right? So let's say we're just using some human cell line that's commonly used to study DNA damage repair processes in mammals. So let's say we have 3 batches of cells. Now what we can do is expose the cells to different amounts of UV light. So let's say we have 0 minutes of UV light, and that will sort of be our control for what the cells, what the levels of CSA messenger RNA levels are in normal cells when they're not exposed to UV light. Let's say we have 1 minute of UV light and 2 minutes of UV light. So 3 samples of cells. Then what we would do is, after exposure of the cells to UV light, we would need to isolate the RNA, right? Isolate RNA. And I won't write out isolate RNA for each little arrow here. You know what I mean there. I'll put little quotation marks instead. So isolate RNA for each one. And then we're going to have RNA samples for each treatment there. So what we need to do now is, I'm going to go down here. I'm going to use those images in a moment, those diagrams in a moment. Now what we need to do is separate the RNA by gel electrophoresis. And we've learned about this process. Usually we've learned about it with respect to DNA, separating DNA molecules by length using gel electrophoresis. So we can do the same thing with RNA molecules. So let's say that's our agarose gel. Now with RNA, we are going to need to add something to the gel to keep the RNA molecules from folding into strange shapes. And why does that happen? So a common molecule that we will add to the gel is going to be formaldehyde. Now, RNA is single-stranded. Most RNA in eukaryotic cells is single-stranded. And messenger RNAs will have a poly A tail, 7-methylguanosine cap. And even though I have it diagrammed here as a single strand, if we don't have, say, a chemical like formaldehyde in the gel, what can happen is that RNA can fold into strange shapes by intramolecular hydrogen bonding. So little sections of the RNA that happen to be complementary to each other can fold, and we get these strange secondary structures forming in that RNA. And when that happens, well, the RNA molecules would separate by length and shape too, right? So we wouldn't really get a clean separation of the molecules based on length alone. So by adding formaldehyde, we keep that intramolecular hydrogen bonding from forming between various parts of the single-stranded RNA molecules, and we can separate the RNA molecules by length alone. So let's say we load the 3 samples up here in our wells of our gel. We separate from long to short. The shorter molecules are moving faster than the longer molecules. And, okay, say the RNA has migrated through the gel. We loaded the 0 time point in here, 1 minute time point, and 2 minute. 1 minute in the middle, 2 minute on the end. And if we added, let's say, a stain that bound to the nucleic acids, and we took an image of the gel, well, we probably won't be able to see most of the RNA. So we might see, if we had a ton of RNA there, we might see, some smears. But eukaryotic cells make a whole lot of a few RNA molecules, right? The ribosomal RNAs. So eukaryotic cells have tons of that. So we should see some pretty intense bands corresponding to the different size ribosomal RNAs. So I'm going to put 4 in here. And so what would these be? We talked about the ribosomal RNAs quite a bit in this course and in BSc 219. So this is going to be, the 28S. Then we'll have the 18S ribosomal RNA, S being Svedberg, and then 5.8 and 5 down there. So we might be able to see these. But we won't be able to see really individual messenger RNAs because the levels just aren't high enough. So in order to, in our hypothesis, remember, was, that CSA messenger RNA levels go up after exposure to UV light. Okay, so we need a way to detect the CSA messenger RNA molecules in this after separation by gel electrophoresis. So what we need to do is we need to transfer all of the RNA to a solid support. So let's say the semi-solid support is the gel. So all the RNA is in that gel, but that gel, it's like gelatin. It's like Jell-O. So you have to be very careful with it, and we can't really detect specific RNA molecules when the RNA is in the gel. So we need to transfer that to a solid support. And the most common support used for northern blotting is a nylon membrane. Now this is a little bit like Western blotting, and I think with Western blotting we normally use a nitrocellulose membrane, so it's very similar, where we need to transfer the proteins from the gel over to a solid support like a nitrocellulose membrane. In this case, we're going to use a nylon membrane. A nylon membrane is positively charged. So it binds to the negatively charged RNA molecules, which are negatively charged because of that phosphate backbone. And essentially a nylon membrane, essentially it's like a sheet of paper that does not dissolve when it's wet. So it's very similar to a sheet of paper that does not dissolve when it's wet. So it's a sheet of paper made of nylon. And it's porous, so things can flow through it. Solutions can flow through it, but the RNA will stick because of the charge differences there between the RNA and the charged membrane. So there are some things we're going to use or need to transfer the RNA from the gel to the nylon membrane. And this is a very, low-tech process, but it works really well though. So I'm going to go up here to my diagrams. So we are going to need a couple of different things. So we are going to need a tray of buffer. And the buffer, that blue stuff right there, that buffer, that's going to be high salt buffer. Let me save this here. I better save this in case I screw up this video, because I don't want to lose these diagrams and have to make these again. So we have a tray here filled with buffer, with the high salt buffer. And one of the nice things about the salt, it helps maintain the charges on the nylon membrane and the charge differences there. But also it helps inhibit enzymes that might degrade the RNA. So, and the buffer is going to help transfer the RNA from the gel to the nylon membrane. So we have this tray with a buffer. So this is going to be our nylon membrane. This right here is going to be some blotting paper, which is, just like a really thick paper. It doesn't tear easily when it's wet, and it's really absorbent. And here we have a bigger sheet of blotting paper. And here we have a sort of solid block. And it can be plastic or any material, really. Here we have some paper towels. And here we have our gel. And our gel I have it diagrammed as lying flat. So the gel where the RNA has already been separated by electrophoresis from long to short molecules. Now the nylon membrane and this little blotting paper right here, they both have been cut to the size of the gel. The paper towels have also been cut to the same size. And what we're going to do here, essentially, we are going to put this solid block into this tray of buffer. We're going to put this long blotting paper here, this big piece of blotting paper, over the block. And these ends right here are going to hang in the buffer. So the buffer is going to flow up into the blotting paper. And it's not going to stop there. Why isn't it going to stop there? Because we're going to put the gel on top of the blotting paper. Then we're going to put the nylon membrane. We're going to lie it nice and flat on that gel. Then we're going to lie this blotting paper on top of that nylon membrane. And then we're going to lie these stacks of paper towels on top of that rectangular piece, single sheet of blotting paper. And this blotting paper, this might not really be necessary, but it's nice to have something that's a little thicker and doesn't really dissolve and fall apart when it's wet in contact with the nylon membrane, rather than just putting paper towels on the nylon membrane. So this setup essentially is going to just drag the buffer through the blotting paper, through the gel, and it's going to carry the RNA molecules from the gel onto the nylon membrane as the buffer is flowing through the nylon membrane, through the blotting paper, up through the paper towels. So the setup, when it's assembled, is going to look something like this. Oh, and I forgot to put, we have some sort of weight. It's very common to use just a book in the laboratory. Just put something heavy on top of the paper towels to keep everything kind of, not too squished together, but to keep everything lying flat on top of each other. All the different components of this apparatus here. So we have the tray with buffer. The buffer's flowing up through the gel, and it's dragging the RNA molecules with the buffer flow. So it's moving, the buffer's moving by capillary action because the blotting paper absorbs the buffer, and the paper towels absorb the buffer as well. So the buffer's moving up through, and it's carrying the RNA molecules through up onto the nylon membrane. And then RNA molecules will stick to the nylon membrane. So that sits for about 24 hours, typically. And then after that, okay, so what do we do? Essentially, after that, let's see. We have to take apart the apparatus here. And I'm just going to focus in on that gel here. So here was the gel with our lanes where we loaded the RNA samples. And they separated. Now, let me put the nylon membrane on top of it. So there's the nylon membrane that was lying on top of the gel, flat. It's like a sheet of paper, right? Sheet of nylon, thin sheet of nylon. So, okay, the RNA's already moved by capillary action. The buffer flow was flowing up. Okay, RNA moved up with the buffer, but the buffer kept going through into the paper towels, but the RNA molecules got stuck. So after about 24 hours, what we do is we peel this off, and then we have our nylon membrane with RNA molecules on it. And you know what? We can cut a little notch in the membrane. So let's say this is our gel here. We can put a little notch in the nylon membrane so we always know the orientation of the nylon membrane relative to the gel. Okay, so there's our nylon membrane with RNA. So we can't see anything though, right? It's got all the RNA, it's got the ribosomal RNAs, and we could stain it with a stain that binds nucleic acid, randomly, and we would see the ribosomal RNAs again, but nothing else really. We want to see, where we want to detect the CSA messenger RNAs, because that's our experiment, right? We're testing for levels to see if the levels of CSA messenger RNAs go up. So what we're going to use, what is most commonly used, is a radioactive probe for the messenger RNA. So let's see, what color do I want to use? I'll use blue. So there are a number of different type of probes we could use. We could use a single-stranded DNA probe or a single-stranded RNA probe. A single-stranded RNA probe is the most common. So our probe will have a 5' end and a 3' end, and it will be complementary to the CSA messenger RNA. And there are a number of different ways we can synthesize this. Just know that the probe we are using is complementary to the CSA messenger RNA. So if these are, let's say, and the probe can be long, it can be a couple hundred base pairs long, it could be 1,000, not base pairs, bases long, or nucleotides long. But I just want to point out that these phosphates here, now these are going to be radioactive. If we're going to use a radioactive probe, we're going to use, when we're making the probe, we're going to use radioactive NTPs that have that phosphate that is closest to the 5' carbon. It's going to be radioactive, and it's going to be radioactive with an isotope of phosphate called 32P. So this is an unstable isotope of phosphorus, and technically usually only 1/4 of the phosphates would be radioactive, but that's a detail that we can ignore. So let's assume all of these are radioactive. They're all the 32P version of phosphorus. So 32P breaks down. It has a half-life of about 2 weeks, and it breaks down into 32 sulfur plus a high-energy beta particle and high-energy electron, which is considered a beta particle because the way it breaks down is called beta decay, and an antineutrino. And that detail isn't too important. This isn't too important. What's important is this thing right here. So this is what we're going to use to detect where that probe is found. This high-energy beta particle that results when P32 breaks down. So after we make our probe, and the details of how it's made aren't really important, we won't cover that in this video, but we have our probe, which is radioactive, and it's complementary to the CSA messenger RNA. So what we would do is, there are a number of ways to do this, but we have our nylon membrane, we have our probe. Oh, I forgot one thing. So we have our nylon membrane with RNA. Usually we are going to cross-link the RNA covalently cross-link the RNA to the membrane by placing the membrane under a UV light. And that will cause covalent bonds to form between molecules in the nylon and various parts of the RNA molecules on that membrane. So that just allows us to use really high temperatures if we wanted to when we're trying to get this probe to stick to the messenger RNA. So that's a detail that I added. It's not really that important. So let me get rid of that, clean this up a bit. Okay, so here's a tray. And what we're going to do now is we are going to put our nylon membrane in this tray with what is called hybridization buffer. So it's a buffer that's going to allow our probe, there's the buffer, it's this blue color, and the nylon membrane is submerged in the buffer. The buffer is going to allow our probe to hybridize to any complementary RNAs on the nylon membrane. So, and then this might be on a shaker, and there are many ways to do this. So often these are done, this is done in a bottle, a hybridization bottle, or like a Ziploc bag or something, heat-sealed Ziploc-like bag, because these are, this radioactive probe, it's radioactive, it's dangerous, so you have to be very careful, using all sorts of shielding and things like that to minimize the researcher's exposure to P32, those beta particles when P32 breaks down. So I have this on a shaker right here. This is one way we could do it. So we'll be adding the probe. We say this is the probe for CSA, the single-stranded radioactive probe. We'll add that, and then we'll shake it we can shake it overnight, usually at a certain temperature. that promotes hybridization to the CSA messenger RNA without non-specific hybridization to, let's say, messenger RNAs that it might be partially complementary to. So we give it time to find the messenger RNAs on the nylon membrane. And then what we do the next day, what we can do is we can see, well, you know, maybe I should explain hybridization a little bit. So what's going to go on? Hybridization, what do I mean by that? So here's our probe with the radioactive, a section of our probe with the radioactive phosphates. I won't diagram the phosphates. Let's say here is part of a CSA messenger RNA that's stuck to the nylon membrane. I won't diagram the nylon membrane, but I'll put the 7-methylguanosine cap here, and then way down here somewhere there'll be a poly A tail, right? And here, okay, what happened? So this is hybridization. The probe is stuck to the CSA messenger RNAs. So after giving the probe time to hybridize to the messenger RNAs, what we can do, or to the CSA messenger RNAs on the nylon membrane, what we can do is we can detect where the probe is using a process called autoradiography. So here is, let's say we take the membrane, we take the nylon membrane out of the hybridization buffer, and there are some washing steps we need to do to rinse off excess probe that's not stuck to any of the messenger RNAs, but we can ignore those. So let's say this is lane 1, lane 0, lane 1, and lane 2. And what we need to do now is put a piece of X-ray film working in the dark, or in a dark room with a light that won't damage the X-ray film. So we put a piece of X-ray film over the nylon membrane, which is damp, so we probably wrap it in a piece of saran wrap. And that doesn't bother the beta particles, they go right through the saran wrap. So we put the X-ray film over there, and then X-ray film usually has a layer of silver halide crystals. I put an X because, you know, the specific silver salt there doesn't really matter. You know, they're different for different brands of X-ray film. So essentially what happens when P32 breaks down, right, breaks down to S32 plus a beta particle plus an antineutrino, the high energy electron, the beta particle, when it hits the silver crystals that are in the X-ray film, it causes them to form into silver, the metallic form of silver, so silver metal. Probably butchering the chemistry here, but we'll leave our physics professors and chemistry professors to, you know, explain more about this process. What we care about is that this silver metal form, this is what gives us the dark spots. So the darker the spot on the X-ray film, the more metallic form of silver that was present on the nylon membrane, the more metallic form of silver, the more P32 decay. The more P32 decay, the more of the CSA probe that was on the membrane, and the more probe that was on the membrane, it means that we had more or higher levels of CSA messenger RNA. For example, let's say the CSA messenger RNA is here, and let's say at time point 0, it looks something like this. You know what? I better change the opacity. And let's say at time point 1, I'm going to change this opacity a little higher. 1 minute time point. The signal's like that. This isn't the nylon membrane I'm showing you. I'm showing you what the X-ray film looks like after we develop it. And at 2 minutes, it looks like this. Okay, so what have I done here? We're looking at the X-ray film, and we're seeing these dark spots, which are light because of the way I'm diagramming this. Oh, you know what? I probably could change this to a white background and actually diagram the spots black, but just imagine that these spots are black and the background is white. So the level of signal here is related to the amount of probe. So here we have more signal than we have here. So it looks like our hypothesis is true. If there's more signal here, it means if there's more signal here, it means there's more probe. And if there's more probe, it means there's more messenger RNA. So it looks like that our hypothesis is true in this case. It looks like when the cells were exposed to UV light for longer amounts of time, their levels of CSA messenger RNA went up. Okay, so that's essentially how we can use a northern blot to, test a hypothesis that is dependent on, that is related to messenger RNA levels. So one more thing. So I need to explain one thing, because we're gonna look at some northern blots when we start talking about, in the next video, when we start talking about exon junction complexes and how they were discovered. So one caveat that we need to discuss is levels of RNA are important. And by levels of RNA, I don't really mean levels of the CSA messenger RNA that we just discussed. I mean the amount of RNA that's loaded onto a gel for gel electrophoresis to perform the northern blot. For example, going back to our last experiment, we have the 0 minute, 1 minute, and 2 minute time points. Now, what if we loaded, let's say, 10 microgram of total RNA for the 0 minute time point? And we loaded 10 microgram of total RNA for the 1 minute time point. And here, let's say we thought we loaded 10 microgram, but in reality, we loaded like 0 microgram. Let's say we forgot to load the RNA in there. So when we go through our experiment and we do the northern blot, we might see something like this. So a little CSA messenger RNA there, a little more here, and then nothing here. So that would change our interpretation, right? That would affect our interpretation. And we wouldn't really know that this is due to a technical error, right? We might not know that. So we need a control to know, sort of estimate or let us know that we loaded, similar levels of total RNA for each treatment. And it's very common to use what is called a housekeeping messenger RNA as a control for a northern blot. And what do I mean by housekeeping? A housekeeping messenger RNA or gene, housekeeping gene is usually it's thought to be a gene that, its levels, its expression levels don't change no matter what the condition. Now, it's debatable whether there are any true housekeeping genes that never change their level under any condition. But one that is often used might be the actin gene. And the actin gene, encodes an important protein that is part of the cytoskeleton. So the actin gene is often used, or actin messenger RNA is often used as a control to let a researcher know that, they loaded the same amount of RNA into each lane. So, and you can include 2 different probes in the hybridization buffer, so you can detect both RNAs at the same time, as long as they're different sizes. So let's say the CSA messenger RNA is 4 kb, and this might not be true. I'm just coming up with some lengths. And the actin messenger RNA, let's say that is 2 kb. Now, if we loaded the same amount of RNA in each lane, we should see the same amount of actin messenger RNA in each treatment for the 0, the 1, and the 2-minute time treatment. Why? Because actin, is usually a gene whose expression levels are the same, no matter the treatment. So if the actin levels are the same, then we can know that, okay, we must have loaded the same amount of RNA into each lane during the gel electrophoresis step. And then we would compare those. Instead of making the bands darker, I'm just going to make them bigger to indicate more CSA messenger RNA. So now we know these differences here are meaningful for the CSA, because when we look at the levels of actin, we know the levels of actin should not change between treatments. If the levels of actin are not changing between treatments, we know we loaded the same amount of RNA. And when we look at the CSA messenger RNA levels and we see that those levels are going up relative to actin, we know that, okay, so it must be the treatment conditions that are causing the CSA messenger RNA levels to increase. Okay, well, I hope you understand how northern blotting works. We are going to look at, I think, a really cool paper, a cool experiment, which helped researchers identify these things called exon junction complexes that are installed on the exon junctions during splicing or after splicing. And they help cells determine, well, they're important for a process called nonsense-mediated decay, which we will also be discussing. Okay, see you in the next video.